Hangout is live. All right. Okay, so we've got a few people logging in asking how to join in. Hopefully we can bring them in. But I can start a long rambling introduction while we find some extra people. All right, so motivation for doing this was partly a couple of reasons. Hi, Ohio State. We've got the Ohio State on the line now, Mike Wilkins. But actually, part of this is I've seen a lot of you in the last year, right? We've been driving and flying to chat with each other, and we'll just stay video and audio. Okay, thanks, Daniel. Anyway, but making lots of efforts, spending lots of carbon to chat with each other. And I don't know about you, but in my home institution, we are a marine biology or marine sciences institute. We don't have that many microbiologists, although we do have a nice quorum. And so I thought, I have a small group, how can I expand things? I've worked in network talks with NASA and also with the Center for Dark Energy Investigations, or CDEBI, doing network talks, and it's a great way to get everyone together, people that you'd see at a conference maybe once or twice a year, and bring them together in terms of sharing information. And so I'm active on social media, as many of you know, sometimes too much. And my colleague Julie Maresca just joined in, and so I can tease her that she always tells me I shouldn't meet people online. And that, what was I going to say? So actually on Facebook, Brandon Briggs had posted that he would like to get experience doing talks and is looking to share some new data that he has. And so I approached him and said, hey, would you be willing to do this in terms of a large networked format? So in terms of open access free platforms, we are doing the Google Plus method. We are trying to tape and broadcasting it on YouTube afterwards, especially for people in other time zones. I have some colleagues in Singapore and Australia that expressed interest in joining in, but of course for them the time zone issue is difficult. And I was wondering if actually Brandon's collaborators from China would be joining us.
Yeah, it's midnight there right now.
Hey, what scientist sleeps, right?
Yeah.
So these are the motivations, and again, everyone's going to be muted during the talk, other than Brandon, and if you have questions, you can type them in the chat window. And I know some people might pop in and pop out, and so we'll just try to ignore that fact and talk to each other otherwise. So any other questions before we start Brandon trying to share his screen? No, we're good? Okay. So Brandon, let's talk about you. You were a postdoctoral or a PhD student at Oregon State University with Rick Caldwell, and had done your thesis work on sediments and hydrates.
Yep.
Right? And so then since then you've moved to postdoc at the Miami University in Ohio. And you've been working in microbial ecology of many regions, including Chinese hot springs, right?
That's correct.
Excellent.
And also I'm a CDEBI postdoc, funded through CDEBI right now as well. So.
Yes, and that was to look at micro-mineral interactions, right?
Right.
Yes. So someday, Sarah and Daniel and a bunch of the other mineral people online, right, I'm sure would like to hear the updates to your research.
Yeah.
Cool. Okay, well, Brandon, why don't you try sharing your screen, make sure it works across the group here.
All right. Can everybody see that?
Yeah, that works.
And do you want to make it such that, hey, we've got Michigan State online too, right?
Yeah,
all right.
University of Michigan just came in.
Oh, University of Michigan. Okay, sorry. Forget. There are actually a University of Delaware and Delaware State, but we often forget that they're up the road.
Yeah.
All right. So Brandon, when you share your screen, do you want to try to make it a full screen view, or?
Okay. You see that?
Yeah, that looks awesome. All right, so what I'm going to do is I'm going to start working through and muting everyone. Yeah, so we still got a few more minutes, but yeah.
Okay. I'll just,
Okay.
get down and start messing around.
Okay. My lab is still getting on board. All right, so I'm going to go through and start muting people. Oh, you muted that. All right.
All right. Want me to get started?
We still got a few more minutes, actually.
Okay.
So do you want to just kind of give us an introduction in terms of how you got involved in this project, and maybe are you going to talk about the pyre in the talk, or?
Yeah, to a degree. Really, what brought me over to start this postdoc is a NSF PIRE, which is a Partnership in Research and Education grant through NSF. This one's led by Brian Headland at UNLV, and Halian Dong, my main advisor here, is who brought me over to start working on these hot springs. This is a large collaborative group. There's 8 U.S. institutions and 6 Chinese institutions that are involved in this. I'll show you some pictures of us traveling over to China, where we had a group of, it was 30 to 40 of us trotting around the hot springs trying to do sampling and trying to keep everybody organized for that. But not only is there microbiologists, there's geochemists, and chemists that are involved with this, so we're looking at all aspects of these particular hot springs. What I'll talk about today is an aspect of that where we're looking at different times throughout the year and how the microbial communities changed with those particular times.
All right, awesome. Well, let's wait 2 more minutes. I see we just got Kentucky on there, Tennessee rather, sorry. UTK, Knoxville, right?
Yeah, Knoxville.
Hi, Jordan.
Hi.
All right, so the deal is we're going to mute everyone other than Brandon, and that means that he's going to pop up to the main screen on the Hangout while you guys are there. So, sorry, my lab just joined in, and oh, you can see everyone. Sorry, you guys just triple tabs on my computer. So. All right. All right. Sweet. All right. So 1 more minute. We'll give just one more introduction in terms of Brandon, who is a postdoc at the Miami University in Ohio. And today he's going to bring us the microbial ecology of Chinese hot springs. So my clock is showing noon. So Brandon, if you want to take it away, it's all yours.
Well, great. Thank you. I want to make sure to thank everybody that's tuned in to this first network talk. Hopefully we can continue doing these for other different types of talks. First of all, I want to give a great big thanks to Jennifer Biddle for her time and effort in setting this up. I also should make sure to thank the NSF Tectron Pyre team, like I mentioned before. It's a large team of people that are collecting various aspects of these Chinese hot springs. And I should also thank the staff at the Yunnan Volcano and Spa. I don't usually get to use spa in a scientific talk, but we didn't use the spa. We were sampling. And this was funded through NSF and NSF China. So really, before I get started into some of the seasonal changes of these extremophiles, I'm going to start at a fairly broad scale. if my slide changes. There we go. Earth. It's an amazing planet whose processes are both affected and is affected by life. Now, many of us are aware of the plants and animals and the role in the environment, but really to understand how life can support, or how Earth can support life, we need to take a closer look. Not just at this ecosystem level, or at the local level, but really we need to go down to that micron scale level. While microbes are usually too small to be seen with the naked eye, their activities affect things like our lives, our food sources, and global processes such as climate. And really, I've always been amazed at how such small organisms can have such a large impact. This is a large area of your research, trying to figure out how microbes are assembled into communities and how these communities affect different ecosystem processes. In addition, how do the conditions of that particular ecosystem feed back to the microbiology and how they affect the microbial communities? So really a fundamental question to understand this particular loop is how do the microbial communities, how are they distributed, and how does this occur with different seasons? Now, we're very familiar with how certain plants and animals change their appearance or behavior with different types of the year. Say, for example, a tree that loses its leaves or a bear that hibernates during the winter. But really, what do the microbes do? Do they change their activities? Do the abundance changes? Do they show seasonal changes? Well, we have an idea in several environments, such as oceans, that they do show these changes with different seasons. But there are several environments where we don't really have an understanding of this fundamental question. One example here is a hot spring, mainly because, well, a hot spring is where water has been heated through geologic processes and expelled upon the Earth. And because of this, we have a assumption that because of these geochemical processes are created or developed by deep Earth processes deep within the Earth, not by the orbit of the Sun, that they really wouldn't change with the season. There has been some studies despite this, but mainly the assumption has been that light intensity would be changing. And so they've looked at photosynthetic mats and seen how the light intensity would change. And so they've looked at a comparison between a tropical, where we really don't see much change in the solar radiance, versus a temperate, where we would have much larger changes in solar radiance. And these studies have used methods like DGGE and clone libraries. And really what they found is something kind of interesting was in the tropical environment, these phototrophic mats actually changed with the season. There were differences in some of the geochemistry as well as some of the microbial communities. When you go to a temperate environment, such as Yellowstone National Park, they didn't see these particular types of changes. The location might change a little bit where these mats are, but the microbial communities really didn't change. And even when they artificially reduced light intensity, they still didn't really see a change in these microbial mats. So really there's a large knowledge gap. There's only been a handful of these studies, and such as, you know, what type of microbial communities in the water and the sediment, not just the microbial mats, how are they changing? Can they change with different seasons? What if we go above 73 degrees, where photosynthetic life is no longer can be supported? You know, can we change those? Does the seasons change those particular communities? And what happens if we use a high-resolution molecular analysis to really dive into that much more rare biosphere than, say, DGGE or clone libraries can? Does the rare biosphere also change? And of course, there's only been a handful of these locations. Like I mentioned, Yellowstone National Park is where most of this has been done. There's been some done in the Philippines and even in the Colombian Andes. But there's only a handful of these locations, and so it makes it really hard to extrapolate out to global environments. So with these knowledge gaps in mind, what we did is we took a trip across the Pacific. I wish all flights were that fast. To China. So notice we have India being pushed up into Eurasia here, creating these Himalaya mountains, and the Tibetan plateaus here. Then over here on the side, in southern China, we have these folds that are being created because of this tectonic environment. And we get a lot of geothermal activity in this particular area. This is Yunnan Province and this Tengchong in particular. I'm going to show you one particular area here, Ruhai. This is a national park. It gets about 500,000 visitors a year. It's granite hosted. We're going to go to a couple of these different springs. This is an acidic spring, 2.5, kind of small. Temperature range between 50 and about 68. Not too far from there is a circumneutral temperature or spring. Temperature is about 98. This is one of our hottest springs. And then if we go down the mountain, by the way, we had to hike up that each time we wanted to do more sampling, is another acidic spring. This is pH about 4.5 and temperatures about 70 degrees. Not too far from that is Guminchen, which had a very high flow velocity, also a circumneutral pH. And then just down from that was this pregnancy well. They would have people drink from that if they wanted to get pregnant, although it did have arsenic in it. And we go all the way down to the bottom to this particular site, circumneutral, high temperature, had these really nice white streamers. We're going to go to another area, not too far, but this is more of a carbonate-hosted system, so a different chemistry. This particular site, Gongoshe, was actually right within the city. This is looking outside my window of the hotel room I was staying in. This was since it was a carbonate hosted, the pH was closer to 9 to 10, also a fairly high temperature in the 80s. This was just kind of a smattering of the different types of springs that we went through and sampled. And one of the first things that we did was, the question is, what kind of microbial communities are here? There really hasn't been much work done in these particular springs. So we went through, collected the samples, and did just some pyrosequencing on the various samples. And at a broad scale, we noticed that really these communities are clustering based off of both temperature and pH. So in this group 1, we had a high temperature, neutral to high pH, was dominated by a particular Ocophaceae. If we go to group 2, which was in that second area, Rudian, within that city, this was very diverse. Most of this was biological dark matter. We only know of it because of the sequences that we were able to get from there. We don't really have a whole lot of cultured representatives from this particular area. And then we had 2 different groups here. Both of them had that low pH, pH around 2.5, but they separated out by having a low temperature, or between 55 and 64 degrees, or a high temperature around the 85 degrees. The low temperature here had another Ocophaceae, a different genera than what we found up in group 1, and the high temperature had a lot of crinarchaeotes in there. So on a broad scale, we're just kind of seeing that these form very nice distinct groups, and that temperature and pH had the highest correlation to these particular groups. Like I mentioned before, we kind of interested in, can there actually be seasonal changes? Well, Tengchong, it's a tropical area, has 2 seasons, a wet season and a dry season. In the winter months, it's fairly dry. You don't get a whole lot of precipitation. But then in the summer, we get the monsoons that come in, and we get quite a bit of precipitation. Trust me, I was in most of that rain. The solar radiance really doesn't change a whole lot throughout the year. I'm going to go through and explain a sampling in January, also one in June. I'm going to briefly talk about a sampling that we did in August, but mainly I'm going to focus on this comparison between January and June, between a fairly dry and a fairly wet season. So several people have gone through and measured the geochemistry from right next to where we sampled the microbiology, so we were able to see how the geochemistry is changing. A typical question is, if there's going to be changes in the microbial communities, there should also be changes in the geochemistry. So what I'm showing here is a hierarchical clustering of sites that are based on the change in geochemistry from January to June. So these black ones increased in June, the white ones decreased in June. Some of the gray ones really didn't change much. If we look at the ones that are increasing, some of this potassium, calcium, sodium, dissolved organic carbon, a lot of this stuff is typically higher in the surrounding soils and the environment than a hot spring, and it seems to be increasing. If we look at some of the stuff like iron and sulfate, they're typically decreasing. These are typically higher concentrations within the spring itself, and they're decreasing in June where we get that higher water. So this is really our first inclination that we might be getting an input from rainwater, that really these springs are not independent from their surrounding environment. But this is the geochemistry, so I'm going to move over and start talking about the microbial communities. And to measure these, we use the phylochip, which is a microarray technology. It has probes on it for 16S RNA genes. The latest version that we were using, the G3, has almost 60,000 known taxa, probes for known taxa, with a little over 1 million different probes. So each of those taxa has redundant probes on there, and there are select changes, so we can tell not only what taxa is there, but some of the closely related taxa from that particular probe. This has an advantage because we're putting our DNA on there, and the detection of this DNA is not dependent on its concentration. We can dive much more deeper into that rare biosphere than, say, some of the sequencing methods where we would have to sequence a lot more to be able to get to that same level. Of course, the down advantage is that we're only going to be able to detect the taxa that are on that particular probe, or the probes that are on that particular microarray. So we went through and we were actually using this. We were able to identify an additional 54 bacteria and 11 archaeal phyla than what we did with the pyrosequencing earlier that I mentioned with the +1 paper. So we greatly increased the biodiversity that we knew of in these particular springs using this. And I should mention that the pyrosequencing, we had a fair number of reads, 10 to 15,000 reads per site. So we could probably do a little more sequencing, especially if we started using the Illumina platform. We can get more into this rare biosphere and detect more of these phyla. We were able to detect with this phylochip almost 5,000 different taxa, which is not as diverse as, say, a soil sample, but it's fairly diverse for hot springs. So let's dive into the results of that. First, I'm going to show you the community structure for January. What this is a non-metric multidimensional scaling where samples that are really close to each other, they have similar or very closely similar microbial taxa within that particular site. Ones that are very far apart from each other had very different microbial taxa. This is all based off of the phylochip detected taxa. In January, we found 3 very nice distinct groups. Again, based off of temperature. So groups over here had typically a higher temperature. pH, this is iron. These are the low acidic sites, which is co-correlated with pH. So all these had higher iron. The reason pH is not significant in this, because we do have 2 of our very high temperature acidic sites over here. So pH isn't significant because we do have some low pH environments right here. And then this group 2 was mainly from that second site, Rudian, which was carbonate hosted and had more magnesium in those particular sites. But we did see that temperature had the highest correlation to this community structure with an R² of about 0.36. Now if we contrast this to what we found in June, we're not finding these very nice distinct groups. It was very hard to actually point these out, pull these out of there. And we're actually seeing a little difference in the geochemical control, or at least the ones that are correlated to this community. Temperature is no longer significantly correlated. In fact, pH becomes a significant correlation with an R² of 0.54. And some of those high temperature acidic sites, they're found a little bit closer to those other acidic sites in this particular plot. So the community structure is changing. There's something that's either obscuring the true community structure, or really it's just a crapshoot that they're all just kind of intermingled within this particular time sample. So we wanted to take it a little bit deeper and take a look at what actually microbes are changing for these 2 different times. So what I did was a conical correspondence analysis. Really, we constrained that first axis to show the variation in time, January versus June. All these, the square or the crosses and the circles are taxa that have been detected. The circles are archaea, the squares are bacteria. So everything over on this left-hand side was found either in higher abundance of January, or if it's circled, they were only found in January. If it was over here on the right-hand side, found in higher abundance in June, or only found in June. If you notice very quickly, all of the archaea that was found in June were also found in January, but there were much higher abundance in January. The abundance seems to decrease in June. We also, in June, had a much higher diversity of microbial taxa found there. And if we start pulling out what these actually taxa are, most of them are these soil or mammalian fecal-associated taxa. Taxa that we would not typically consider to be adapted to these high-temperature environments. They're more of a low-temperature or soil-type environment. So if we go back, and we refer back to this NMS here, where we had these, just kind of a random assortment, and we're getting these more of these soil-type microbes. If rainwater is coming in, bringing external non-endemic microbes into the rainwater, that, and putting them into all of the springs, that there would start to obscure this community structure. So we're really starting to see that these hot springs are not independent from the environment, and that maybe these seasonal changes that we could be seeing are simply because of the rain washing in certain types of microbes. Let me briefly talk about August as well, where we're seeing the same sort of thing, where January, these are actually Illumina sequencing OTUs. January there was 80, in June there was 87, so a few more in there. But then in August there was a whole lot more of OTUs that were only found in August, 285 OTUs. And just remind you, August is near the end of this rainy season. So if there was microbes and they're just constantly being delivered into this hot spring, we would see more in there. So the question comes, where are these additional microbes coming from? So this last sampling trip, what we did is we thought, well, if rainwater's coming in, we should look at the soil and see what type of microbes are in there and do a comparison between that and the actual hot spring. So this here is Guminchen. It's a high velocity spring. There's a lot of water going through that. There's soil right near here. Here we are removing some of the vegetation and collecting the soil. That was pretty close to that. There was really no conduit, visible conduit at least, of rainwater washing into this particular spring. So we went through and did a daily sampling for 5 days of this particular spring, and then sampled 2 soil sites. I'm going to show you another NMS here where for this particular site we're not really seeing any differences. Through those 5 days of having high rain and low rain events, there was a little bit of variability in the community, but really not ever going close to what we found in that particular soil. And of course, this was a high velocity spring, and so if there was anything going into that soil, maybe it's not actually, or coming in from the soil, maybe we're just not being able to see it at that particular time. So we also went through and sampled a acidic site. DRTY stands for Jiranchu. This was a small pool. This is a 1 liter bottle. It had about 50 milliliters of water in there. And it was situated right underneath this cliff. So this is looking down underneath, or looking down from the top of the cliff. And what we did is took a soil transect away from that particular cliff. During the high rain events, we can actually see this erosional feature, and we can see the water going through this particular channel and dropping off of the cliff and into this particular spring. So this one had a visual. We can actually see the rain going into this. And here's the actual spring here. Most of it is just kind of bubbling up. It's very acidic, so it has a sulfuric smell to it. We did have to be careful walking around there. There was a lot of these little springs in this particular site. And there's a lot of other debris, organic material coming in, these leaves that are coming in from there as well. So interestingly, when we looked at the community there, we saw 3 different groups. One was the soil, which was expected, and this is the transect away from that spring up above the cliff. When we had a low rain event, the spring was typically at 69 degrees, and it had a low volume of water, 50 mils or so. And that formed a nice tight group over here on the right. When we had those high rain events, we can see water going into it, the temperature decreased to about 48 degrees Celsius. The pH remained the same, about 2.5 for both of these. And so there was this one time period where we were able to sample on both before and after of this high rain event. And interestingly, this community quickly recovered within 24 hours, went back to this original community structure. If we look at what microbes are in there, the soil, we found these typical soil ones, the actinobacteria, acidobacteria, and some of the proteobacteria. If we looked at the high rain event, we had some of the same proteobacteria. We also saw some cyanobacteria. They're actually being, both of these were the dominant phyla during this high rain event. Interestingly, 69 degrees is pretty close to where photosynthetic life can actually still do photosynthesis, 73. So maybe dropping that down to 48 degrees allowed for cyanobacteria to grow up a little more. In this low rain event, the Ophicales and the Crinarchaeota, these were ones that we would typically consider to be a hot spring type microbe that are adapted to these hot environments. And so we wanted to take a look if really the abundance of these are decreasing with increased rain, or that they're being diluted out. You know, why is that? The pH isn't changing, the temperature is changing. So I can run another NMS, but instead of this time, I'm just going to show you the genera, or the taxa, that fall within the Octophocaceae and the Crenarchaeota. And so there's this Hygenobaculum, a genus within Octophocaceae, found over here. That's the ones that are typically found in these acidic environments. We have the Hygenothermus and Hygenobacter found over here in these more alkaline type environments. And pH was the most significant variable that correlated with this distribution of the Octophocaceae. Temperature was not significant within this range that we measured between 50 and 93 degrees. We also notice, well, there's some of these other ones, iron and the sulfate, that are co-correlated with pH. But also this phosphate, ammonia, and nitrate, Hygenobaculums found along these, which indicates that they can handle a variety of concentrations of these particular elements. In Crenarchaeotes, again, pH was a significant variable. Sulfobales, they're typically found in these low pH environments. Then we have some of these thermoproteales, disulfuricales, found over here in the more of the high pH range. Interestingly, I do want to point out this candidate division SK213. We found one over here in a very high pH greater than 9. Sorry, I should point out these contours are actually pH contours. So if it was found on this other side of 4.5, it was just slightly the pH of this particular taxa was found typically in a pH less than 4.5. And so this SK213, we had one found in a high pH and one in a very low pH. I suspect with higher sequencing resolution, this particular candidate division will split up into more divisions. And so if the pH wasn't really changing, why would these two particular ones decrease with lower temperature in this particular high rain event? Well, we should keep in mind that this was based off of Illumina sequencing and its relative reads. So we really, these other ones may have increased in relative abundance, but these may have not have changed at all. So in summary, you know, the phyla chip was able to detect a significantly more phyla than any of the previous reports, especially in this Tengchong environment. We are seeing some temporal differences, not only in the geochemistry, but also in the community structure, but likely because of this rain event pulling the soil microbes, these non-endemic microbes, from the soil and putting them into the particular hot spring. So there's a number of questions that still remain that we're actively trying to pursue, such as, really, does the abundance of the Octophocaceae and the Crenarchaeota change? So we're going through and doing quantitative PCR of these through this time series. Also, are the non-endemic microbes active? Are they actually surviving in this environment? Or if they are, how does the change in the activity of the endemic microbes, how are they changing the activities during these high rain events? And so we're in the process of doing a metagenomics and metatranscriptomics analysis of these particular springs. So understanding the microbial distributions and the effect of environmental conditions, in this case wet versus dry, it's really a necessary start and leads us to the next step in understanding how activities are altered, which then can lead back to global cycles. And so with that, I would like to thank everybody for your attention, and we can try taking some questions.
Thanks, Brandon. That was awesome.
Thank you.
Clap.
So I am.
Or just like he's clapping.
I'm muting Daniel on there loud.
Hot sea, big spring.
All right. So
Do you want to? What?
Who's moderating? Is that
Jen. Yeah. Do you want to ask a? I mean, I can put us like video.
Your mic is on.
Hello.
All right, let's go to Minnesota. Do you guys have a question?
If they're missing anything with phylochip, then they would see this experience.
Well, Julie just posted. Did you ever analyze the microbiome in the rainwater itself? Yes, we do have samples of that rainwater, and hopefully this next Illumina sequencing, we'll be able to get that to work. We ran it before, and something happened, and we weren't able to get the actual results from that. So stay tuned. Hopefully, we'll be able to look at that as well.
All right, it said there's some more chats coming in, but why don't we just have everyone raise their hand in the chat room to ask a question, and we might have some questions coming in from Twitter also. Do you guys have any questions? Yeah, here, come talk. Sorry, we've got a bunch of people in the room here.
Hey, Bren. Hey, I was wondering, do you know the people that live in that area? Do they, during the cold seasons, or do they normally use something like coal or wood to heat their homes? I just didn't know if that might contribute to like acid rain. Maybe that could affect pH, soil runoff, things like that.
Yeah. So as far as China is concerned, this is a fairly pristine environment. Although there's still Yunnan, not too far away. There's Tenchong, a couple million people there. It's not like Beijing's air quality. So we really don't see much of a difference in the pH. The pH of the rain, when we measured it going through that soil had a lot of sulfide in it just from that particular spring. And so the spring water or the rainwater is going through that soil, picked that up. Before it even got to that spring, it was still a pH of about 2.5.
All right. Thank you.
Cool. All right, so you've got a question from chat from Daniel Bond, or someone at Minnesota. There's apparently 10 people in the room there. Says, we were wondering if you could tell the low temp community came from the soil or was clinging to life in the spring, waiting for the temp to drop. Also, Lori wants to know how much you have to drink from the well to get pregnant.
If you could tell the low. Yeah, so I think the proteobacteria that we're finding in that particular spring did come from the soil. We're going to go through and do a little more of a phylogenetic analysis, and so we can tell just how different phylogenetically those proteobacteria were. Based off of the classification, they were down to the same classification. So I would suspect those are coming from that particular, from the soil. The cyanobacteria, we really didn't see those in the soil all that much. And so they might just be in there kind of clinging on to life and hoping for that temperature to drop. This particular spring, in the high rain events, it comes quite often. So I can see them just waiting for that particular right condition and then growing greatly. And how much to drink? I think it was just supposed to be a cup, but there was a lot of arsenic in there, and so I wouldn't recommend it.
All right, so speaking of arsenic, Mike had just asked you via chat that you mentioned arsenic, and now you've mentioned it twice, so it has to be real, right?
Yeah.
So is there evidence for arsenic transformations in these systems? Have you seen anything specifically for arsenic processing?
Yes, and so we've gone through and did this PCR for the arsenic reductase gene. I'm forgetting now. Gung Wu at the China University of Geosciences in Wuhan has been doing that, and they do have some of the genes. Some of the elemental mapping and stuff that we've done, we really haven't seen a whole lot of that. So right now I can say that they seem to have the potential to be able to do some of the transformations at least.
All right, we had a question come in over Twitter. Has any of the geochemistry data shown any changes that might suggest changes in function for the microbial community in August?
So we're still kind of looking at the geochemistry from August. Basically, we're still seeing the same sort of trend with more of the soil type of geochemistry coming into the springs. There seems to be a decrease that might have been because of at the beginning of the rain event, we're going to get a lot more erosion, or picking up more of the erosion, but then that might die off a little bit. There are people that are looking at these as far as rates. We've been trying to do more of a carbon and nitrogen rate measurements. Those are still pretty preliminary in trying to get the kill controls to work in the right way. So I'd say stay tuned for the rates. That's kind of where we want to go with this.
All right, does anyone else on the chat or on the internet have a question for us? I don't see anything else coming in right now. So, oh wait, oh, no, we're good. All right, well, I think what we'll do right now is thank Brandon for his bravery in being the first person to do this. And if you had a good time, please email us and sign up to give a talk and tell your friends and neighbors to join us next time. So thanks, Brandon. We'll give you claps. I think there's an app to do that, possibly. And we definitely appreciate you being the first one to run through this.
Well, thank you. I appreciate you getting this set up and letting me be the guinea pig.
No problem. All right, here, we'll do the Google Effects claps for you. Congratulations, Brandon. At least you didn't hear this.
Yeah. Yeah.
Anyway. All right, thanks, Brandon. That was awesome.
Thank you.
Thanks, Brandon.
Bye.
Bye.
Bye. Minnesota.
Oh, there's a large group there.
Killing me. Look at Jeff. Jeff, put your phone down.
Yeah.
Back to mute.
All right, bye, guys.
Yeah.